Well, hello and welcome back. In this particular lecture, I plan to go over one of my favorite topics, which is blood. As you can see in this particular picture here, this is what blood looks like under the microscope, and you can see that there are different components to it. You have, of course, the plasma, which is this clear liquid portion. There is a there are cellular components, including red blood cells and white blood cells. And then you can also see cellular fragments here called platelets. So during the course of this lecture, we'll be talking about plasma, we'll be talking about the cellular components, and then also platelets. So blood is a connective tissue. It basically has a matrix. That matrix is called plasma, and it's a very, very important substance. It transports all of our food, oxygen, nutrients, electrolytes, cellular waste products, and hormones. It also circulates disease-fighting chemicals. These disease-fighting chemicals are called antibodies. These are proteins that your immune system cells, called B cells, can actually make, and then those antibodies will target toxins, venoms, or other things like bacteria or viruses. It will target them for destruction. Not only does the blood circulate disease-fighting chemicals, but it also circulates disease-fighting cells, and these are termed your white blood cells. There are many kinds of white blood cells that we'll study later on. So blood is very important in maintaining a steady state, a homeostasis. It also circulates cell fragments called platelets, and these will aid in maintaining the integrity of your circulatory system by plugging up any blood vessel that's bleeding. An underappreciated importance of blood is that it circulates or distributes heat through the body, and it helps to maintain proper pH of your fluids in your body. So when we take and study blood, we can take a tube of it out of your body. So here we've taken and we've done some withdrawal of the blood. We put it in a test tube. This test tube probably has a chemical inside of it that helps to cause the blood cells not to stick to one another. We can centrifuge the blood sample, and when we centrifuge the blood sample and look at it, centrifugation will take and uses gravity, the spin of this rotation, to separate out the heavy components compared to the light components. So the heavy components are going to settle at the bottom, the lighter components will settle at the top. You can see that there are three distinct layers that you can see. We can see a kind of a straw-colored layer here that makes up what we call plasma. 55% of your whole blood is going to be plasma, and it's going to be the liquid portion of your blood. Now, suspended in that liquid will be proteins, electrolytes, gases, nutrients, and then, of course, the main component is water. There is a smaller little divide between the plasma and the erythrocytes, and that's called the buffy coat. The buffy coat is kind of a light-colored coat or layer, and it's made mainly of the leukocytes, which are white blood cells, and also platelets. And then a large component of the spun material are going to be erythrocytes. And so if you take all of the cellular components, this is going to make up what we call the hematocrit. And 45% of whole blood, or your hematocrit, is going to be made of cells. So as I said just a second ago, your composition of blood will include plasma, and it's going to also be the red blood cells, white blood cells, and platelets. And so 55% is going to be plasma, 45% is going to be the hematocrit, or the cellular component of the white blood cells and red blood cells. So let's deal with plasma first. The largest component of plasma will be water, but in addition to that water, 8% of it is very, very important stuff. So the protein portion of the plasma is going to be made of these things called albumins, globulins, and fibrinogen. The organ that produces the largest number of proteins for the plasma of the blood is the liver. So making up about 54% of blood plasma proteins will be the albumins. Again, these are made in the liver, and they function to create osmotic pressure. So water tends to stay in the capillaries because of this osmotic pressure created by these albumins that sit in the plasma of the blood. They literally try to draw water into the capillaries. So if you get an upset of the balance of these albumins, edema may result. So if you don't have enough albumin proteins, you will have edema and your body will swell up. So a lot of times we see in people that have liver dysfunction, we might see edema throughout their body. The globulins make up about 36% of the blood proteins, and there are alpha globulins, there are beta globulins. These particular globulins are made by the liver, and they function in transporting lipids and fat-soluble vitamins. Gamma globulins, which are also termed immunoglobulins or antibodies, these are made by our lymphocytes, specifically a B cell, and they are used to fight diseases. Fibrinogen is a really important component of your blood plasma. It makes up about 7% of the blood proteins. It's made by the liver, and it's involved in forming blood clots. And we'll talk about fibrinogen and how it forms fibrin a little bit later on. So plasma gases include oxygen. This is important in helping cells to metabolize nutrients that we eat. Blood plasma also has carbon dioxide. This is a cellular waste product that's carried by the blood to the lungs to be excreted. And there is nitrogen gas that's in our bloodstream, which has generally no function. Plasma nutrients include amino acids, simple sugars, nucleotides, lipids. So you can have all kinds of nutrients in the plasma of your blood. And then some non-protein nitrogenous compounds might include things like urea and uric acid. These are waste products from the breakdown of nucleic acids and amino acids. These particular products, urea and uric acid, and even ammonia, a little bit of ammonia as well, these are going to be taken in the bloodstream to the kidneys to be filtered out of the bloodstream and made into urine and excreted out through urine. There's also electrolytes that are found in the plasma of the blood. These would include things like sodium and potassium, so calcium, magnesium, chloride, bicarbonate, phosphate, and sulfate ions. These are important in helping nerves to work, muscles to work. They're important in maintaining pH and doing other various functions of the body. I will attach to Blackboard a little video of donation of plasma. So many of you may have donated plasma before to make a little extra money. Maybe that's something you'll do in the future. But donating plasma is an important thing because when we donate plasma, they generally take out your blood. They will harvest the plasma portion of the blood, put the cells back into your body, and from that plasma portion, they will harvest very specific proteins that are used in making treatments for people. One treatment could be a treatment for, say, a disease like hemophilia, so those clotting factors can be extracted out of your blood, and they can be harnessed and then made into a medicine that can be injected into a person that can't form those clotting factors. So I'll put that little video on Blackboard for you to take a look at. So we'll now go ahead and talk about some of the cellular components of the blood. And I like this diagram here because it tells you basically the mother of all blood cells is called a hemocytoblast. This is a pluripotent stem cell, so it has the ability to become many different kinds of cells. These hemocytoblast stem cells are found in adults within red bone marrow, and when these particular stem cells are given certain kinds of hormonal or chemical signals, they will differentiate into various other kinds of stem cells and then into various kinds of blood cell components. For example, if this pluripotent stem cell is given a signal to become a myeloid stem cell, then given other hormones, it can become, so if it's given colony formation units, or CSF, colony stimulating factor E, it's going to become a red blood cell. If it's given colony stimulating factor MEG, it becomes a megakaryoblast, which will become eventually platelets. So if it's given other kinds of hormonal signals, then it can become things like eosinophils, basophils, neutrophils, or monocytes. We'll go over each of those a little bit later on. And if it's given these colony stimulating factors or colony forming units, it can become lymphoid tissue. So given the right hormonal signal, it will become T lymphoblast, B lymphoblast, or natural killer lymphoblast, which will form T cells, B cells, or natural killer cells. So you have to have the right hormone to become, or chemical signal to become a particular type of blood cell. But you can see they all stem from the same, or mother of all stem cells, the hemocytoblast. Again, just a different picture showing you the same thing, the formation of these things from various kinds of stem cells. I'm more used to colony stimulating factors as opposed to colony forming units as the name for the signal. All right, well, let's talk about red blood cells first. So they're also known as erythrocytes. Now, urethra means red, cyte means cell. These erythrocytes are red blood cells or biconcave discs, so you can almost think of them as like a donut without the hole pushed through the donut. Mature blood cells, red blood cells, contain no nuclei. They actually eject it in the process of their formation. 1/3 of the volume of a mature red blood cell is going to be a protein called hemoglobin. This will be the oxygen carrying protein that blood cells are known for. And it has a lifespan of somewhere around 120 days. They're not super big cells. If you look at them, they're about 2 microns wide, 7.5 microns in diameter. Now, in science, we always know that structure is function. So the structure of the red blood cell is related to its function. If you look at a red blood cell, the way that it's shaped and formed, it has a large surface area to volume ratio. So it has a huge amount of surface area compared to the volume that's inside. This allows gases to be absorbed or released through the cell membrane very rapidly. And one of the main jobs of a red blood cell is to in the lungs, to take in oxygen and to release carbon dioxide, and then in the body, when it gets to the tissues of the body, to release oxygen and bring in carbon dioxide. So having a large surface area to volume ratio allows them to do this task very, very quickly. So the way that they're arranged too, it allows them to form stacks. So sometimes you'll see in blood cells, and I'll show you a picture of it in a little bit, blood cells can sit on top of one another and form stacks that allows them to push very easily through blood vessels and be more efficient about going through blood vessels. And then because of the structure, this red blood cell is very flexible. It's allowed to bend and flex so it can fit through the smallest of your blood vessels called capillaries. And here is that rouleau complex, which is a stack of blood cells that are moving through a blood vessel. So this is a very unique structure as function. Its structure allows it to stack in. The function is to allow it to circulate through the blood vessels more efficiently. So red blood cells are chock-a-block full of hemoglobin. Hemoglobin is a super complex protein. It actually has quaternary structure, which means that it's made of 4 different proteins that come together. You can see there are 2 alpha chains, so alpha 1 and 2, and there are 2 beta chains, beta 1 and beta 2. So there's 4 different proteins that come together to form the final functioning hemoglobin molecule. Inside of each of the different chains, there is a super really cool molecule called heme. Heme has in the center of it, it actually has an iron atom, and that iron atom, in the way this thing is configured, makes it so that it very easily attaches to oxygen. It can also attach to carbon monoxide and other gases, but oxygen is the one that we really target with the hemoglobin molecule. So if the iron in hemoglobin is bound to oxygen, it's called oxyhemoglobin. and it has a very, very bright red coloration to it. We know this because we can do an arterial stick. You can stick a needle into an artery, draw blood out, look at the color of it, it's bright red. If the hemoglobin is not bound with iron, then it's called deoxyhemoglobin, okay? And it's going to be a dark red color. Now, oddly enough, one of the characteristics of carbon monoxide poisoning is if you look at the lips of a person, they're going to be cherry red. So if iron is bound to carbon monoxide, it'll be a very bright red color, and it literally changes the lip coloration to make it bright red. It almost looks like the person has chapped lips. So if you ever find a dead person and they have bright red lips, probably it was carbon monoxide poisoning that might have taken them out. So when hemoglobin is not bound with, is bound with carbon dioxide, it's going to form a molecule called carbaminohemoglobin. We'll talk about oxyhemoglobin and carbaminohemoglobin when we do the respiratory system. Now, red blood cells are going to be formed in adults in the red bone marrow, and this is in a process called hemopoiesis, so the formation of blood. There is a hormone that's called erythropoietin, okay, that's going to be the signal to the red bone marrow to produce red blood cells. So the way that erythropoietin is made is by looking at this chart. We can follow that little sequence and see how it is produced. So if you have inside of your body hypoxia or low blood oxygen, there are different organs in your body that can sense that. They have sensors there. So the liver and the kidney are two organs that can sense that low blood oxygen. When they sense low blood oxygen, they are going to produce a hormone called erythropoietin. That hormone will go into the bloodstream. It'll go everywhere in your body. The place, though, that can receive the signal for that is your red bone marrow. And what that hormone will do is it will stimulate an increase in the production, the literal mitosis of red blood cells. It increases the ability of them to mature more quickly as well. So once you have lots of red blood cells that are being formed, then your hypoxia will be diminished, and then therefore erythropoietin will stop being produced by the liver and the kidney. Now, it's important that we don't take too much erythropoietin or get too much erythropoietin, because that could actually form a condition called polycythemia. Polycythemia is where you have too many red blood cells, and it can make your bloodstream get really thick and syrupy. And perhaps if you had too many blood cells, it could stop the flow of blood. Your blood gets too thick and won't even flow through your arteries and your veins. So this is just showing the formation going from the stem cell all the way to the mature erythrocyte. So we have the stem cells that are going to the hemocytoblast that are going to be, committed to becoming red blood cells. We go through a series of stages. We have a proerythroblast, a basophilic erythroblast, a polychromatic erythroblast. We then go through an orthochromatic erythroblast stage. We eject the nucleus, become a reticulocyte, and then eventually, once all of the the messenger RNA in here that's coding for the production of hemoglobin, once all that hemoglobin is made, we then will form the full mature erythrocyte. That will circulate around and until the death of the cell. So this process takes about 1 week to go from the stem cell all the way to the mature red blood cell. 1% of red blood cells are replaced every single day that are circulating in your blood. So in order to replenish that 1% of red blood cells that are replaced, we make about 3 million new red blood cells each second and send those into circulation each second. That is tremendously productive. And the lasting or length of lifespan of the red blood cell typically is about 120 days, and during their lifetime they may travel somewhere up in the order of 700 miles through the body. Okay, so we do have to have a stimulus, which is the hypoxia, to make our target organs release erythropoietin. Once that's done, you're going to have a larger number of red blood cells that are produced, and then we have to have a negative feedback loop shut that down. So again, that's another example of a negative feedback loop helping to maintain homeostasis. So erythropoietin, that's the little code we use, EPO, can stimulate red bone marrow to increase red blood cell formation in about a 10-fold increase. So we're going to go, when we have erythropoietin, we'll go from about a 3 million cells per second to a 30 million cells per second output, which is tremendous. There are many people that when they do sports, they want to try to get an unfair advantage over other people, and they may do some kind of use some kind of performance enhancement. One of the things that some athletes will do, and this is typically people like cyclers or Nordic skiers, something where you have to have long-term endurance. They'll actually take and do two different kinds of things. They'll use recombinant bacterial EPO. So this is EPO that's grown in bacteria and then made into a synthetic EPO that's injected into humans. So and that will increase their production of red blood cells. We do have drug tests that can determine if a person is taking EPO. Some people will do this, so a week or more before their event, they'll take blood out of their body. They will keep that blood either frozen at negative 80 degrees Celsius, or they'll keep it frozen and or in the refrigerator. And then right before their physical activity or their event, they'll put that blood back into their body to increase the number of blood cells they have circulating in their blood. That's called blood doping. Either of these things could be very dangerous and even life-threatening, and also can lead to your expulsion from the athletic event that you're performing in. There have been many people that have lost their medals, trophies, or prizes because of performance enhancement. Again, very, very dangerous stuff. So once all these blood cells are made, we know that they have a 120-day lifespan. So what happens to them when they perish? Well, these red blood cells, can be destroyed inside the bloodstream. That's called hemolysis. They can also be taken out of circulation if they're damaged or somehow defective by your liver, spleen, and bone marrow. There are cells inside of those respective tissues and organs called macrophages. These are phagocytic cells. They have the ability to digest blood cells and the products that come from the blood cells. You do want to recycle things as best that you can. If you look at this blood cell here, when it's broken down, when its lifespan is over, the hemoglobin, so all the constituent materials can be broken down. The hemoglobin molecule, which is about 1/3 of the volume, the globin, which is a protein made of amino acids, that can be directly digested into individual amino acids and recycled. The heme portion is a little bit more complex. So what your body does for heme is it strips the iron. The iron will be then it'll be ferried via proteins called transferrin. These proteins will take the iron to your bone marrow or to your liver. If it goes to your liver, it's stored. If it goes to your bone marrow, it's used to make new blood cells and more new hemoglobin. So the protein portion or the non-iron portion of the heme, it's going to be converted into biliverdin. It's then converted into bilirubin. And then that bilirubin is excreted from the liver into what we call bile. That bile is put into the gallbladder. The gallbladder then excretes it into the small intestine. Okay, I have several pictures because I like these pictures from different sources just to show you where things are happening and all the things that are happening to red blood cells. All right, so let's start here with number 1. Red blood cells are going to die, and they're going to be phagocytosed. They're going to be done in or recycled by macrophages that can be in your spleen, your liver, or your red bone marrow. So the globin portion of hemoglobin is digested into amino acids, and that can be used for more protein synthesis, so you can make new proteins. The heme portion being much more problematic, the iron of the heme is going to be stripped off, and it's going to be transported by a protein called transferrin. That transferrin will take and some of the iron to the liver to be stored. Some of the iron will be taken to the bone marrow in your red bone marrow to be made back into hemoglobin for new red blood cells. So that heme portion, which is problematic, is converted into biliverdin. It's then converted into bilirubin by these phagocytic cells. It's then taken to the liver. The liver will take it out of circulation, store it into, it'll take it out as bilirubin. It'll be stored as bile. That bile will then go to the small intestine. So the bilirubin inside of bile can be converted by bacteria. The bacteria that's normal flora of your intestines can convert it into urobilinogen. Urobilinogen, a little bit of it will escape back into your circulation and form urobilin, and urobilin will be excreted out by the kidney into your urine, and it is a yellow pigment. So the reason that your urine is yellow is because of that, a little bit of that urobilin that is excreted by urine. So urobilinogen can also be converted by bacteria into stercobilin, and that actually is a dark pigment that colors the feces kind of more of a brownish coloration. Okay, so we know now why our urine is yellow and why our feces are brown. It all goes back to the breakdown product bilirubin being taken out by the liver. Now, if your liver's not working correctly, then you can't take bilirubin out. So if you can't take bilirubin out, it will take and cause a yellowing effect of your skin, and that yellowing effect, it leads to a condition called jaundice. So that's a symptom of liver dysfunction. Some babies are born with jaundice because their livers aren't working that efficiently, and hopefully that gets fixed, and if not, surgery might have to ensue. Okay, now I like these other diagrams for different reasons. I like this one. It's a little bit more schematic, a little easier to understand. So, so if we take a look at the red blood cells being broken down, they're broken down into heme. Iron is transferred again back to the bloodstream and its travels attached to a protein called transferrin that goes to the liver or to the bone marrow. The heme portion is digested into biliverdin or converted into biliverdin, and then bilirubin. Bilirubin is then going to make its way to the liver. It is bound to a protein, albumin, and that goes to the blood, and that circulates through the bloodstream, but eventually gets to the liver to be taken out of circulation. Oh, let me backtrack there. So in the bloodstream, sometimes the hemoglobin that's not phagocytosed because you have hemolysis in your bloodstream, that can actually go to and be eliminated by the urine. So if you have major hemolysis, let's say you were bitten by a venomous snake that has hemolytic venom, one of the symptoms will be looking at the urine. You'll actually see some hemoglobin chains inside of the urine. This is just showing you what's happening in the bone marrow. So that iron is transported by transferrin to the red bone marrow, and that can be used to convert that can be used to help to build hemoglobin in new blood cells, which can be put into circulation. And then lastly, this is just actually I have one more. This is just showing you bilirubin bound to albumin is going to be taken out by the liver, excreted by the gallbladder. So it's taken out by the liver, stored in the gallbladder, then it's excreted as bile into the large intestine. That's where the bacteria begin to work on them and can convert your feces into a brown material. but some of the urobilinins can get into the bloodstream, cause your urine to be yellow. And then lastly, we can see that that the kidney can take the urobilinins out and eliminate it as urine. Also, sometimes hemoglobin can be urinated out as well if there's too much of it in the bloodstream. So your red cell counts include, males have about 5,400,000 per microliter. So a microliter is a cubic millimeter, and it's a very, very small unit. It's a thousandth of a liter. Females have about 4.8 million cells per microliter. And when you have too few blood cells, that is called anemia. And sometimes you can also have too little hemoglobin. We also call that anemia as well. Anemia is a broad term that has many different things that cause it. So, for example, one form of anemia can be caused by B vitamin deficiency. So, in order to make blood cells, you have to have folic acid and vitamin B12. Too little intrinsic factor, which is a factor made by the stomach, which helps our intestines to absorb vitamin B12, that can also cause anemia as well. And we call that pernicious anemia when it's caused by too little vitamin B12 being absorbed or that's in the bloodstream. I mean, it's self-evident to you, I'm sure, that bleeding can cause anemia. If you lose a lot of blood cells, you're going to have anemia. Iron deficiency can also cause anemia as well. So if you don't eat enough iron in your diet, that can be a problem. If you have super heavy menstrual flows, if you're a female, then that can cause iron deficiency too. So you be careful with your diet, and hopefully you won't get iron deficiency anemia. So sickle cell disease is a genetic anemia. It basically is an anemia that destroys red blood cells and diminishes the oxygen carrying capacity of the blood in that particular person. So red blood cells of individuals with this disease carry a mutant form of hemoglobin that causes red blood cells to bend into what we call a sickle shape. When it's oxygen stressed. So when these cells are oxygen stressed, they'll get into a sickle shape. They will then get blocked in your capillaries. So you can see normal red blood cells squeeze through because they're flexible, whereas sickle cells are inflexible. The hemoglobin inside actually crystallizes due to lack of oxygen, and then it wedges itself and gets stuck in the capillaries. So everything downstream will now have hypoxia because it's not getting nutrients and it's not getting oxygen. So this is a very painful, horrible condition. This is caused by a genetic mutation. If you take a look at the genetic code, the gene is mutated in one place, and normal erythrocytes, the genetic sequence, actually, when you're building this protein, there's an amino acid that the normal code codes for at the 6th position for glutamic acid, and it's substituted for valine, the amino acid valine. And just because of that one little difference, the hemoglobin's made incorrectly, and this disease results from that. It's kind of interesting. There is a treatment for sickle cell anemia. There is a drug that doctors can give sickle cell patients that stimulates their DNA to produce fetal hemoglobin, which is an alternate form of hemoglobin that you produce while you're a fetus. And that fetal hemoglobin doesn't have the same properties as the sickle cell hemoglobin. Therefore, that sometimes can at least diminish the effects that sickle cell disease has on people. Okay, so there is some terminology that you probably should be somewhat familiar with, especially if you go in the healthcare field. We do have all kinds of tests that we can test for different diseases. So hematocrit, which is HCT, is going to be determine the percentage of the cellular components that are in whole blood. If you have an elevation of that, so I think we said somewhere between 45% or so, it's going to be kind of an average. but 37 to 54% should be what a normal view is. So polycythemia is where you have an elevated hematocrit, and then a depressed hematocrit will lead to anemia. So in some of these blood tests that we do, we'll do a reticulocyte count, which is a red blood cell count. And so if you have too many of them, it's reticulocytosis. And if you have a diminished one, it's called it's because of not enough erythropoiesis or hemopoiesis for red blood cells. So we do a hemoglobin count. We also do a red blood cell count. Reticulocyte is not a completely mature red blood cell, but it is a red blood cell that is maturing. We also do an MCV, which is a mean corpuscular volume. So we can see the volume of a red blood cell. And if it has a large volume, it's a macrocytic cell. If it's a small volume, it's a microcytic cell. And then we do a mean corpuscular hemoglobin count, or MCH, so we can know how much hemoglobin is in the red blood cell. If you have too much hemoglobin, it leads to a hyperchromatic or deeply staining cell, but too little hemoglobin leads to your red blood cells being very light in color. So one day you may order a CBC, maybe you'll be a doctor, maybe you'll be a physician's assistant, or perhaps a lab tech, and you'll either order the CBC or do the testing for the CBC. So a CBC means complete blood count. A lot of times we'll do a complete blood count with a differential where we'll actually see how many different kinds of white blood cells there are and in what number they come in. So this is just a printout showing you what a CBC looks like. You can, if you ever get a CBC, you can ask your doctor for a printout. These printouts usually come with your value. They come with the standard range. They'll come with the units that the test is measuring in, and they'll come with flags if somehow you deviate from the normal amount. So we'll do a white blood count. That's a total white blood count, a total red blood count. We'll actually look at the hemoglobin amount inside of the blood sample. We'll do a hematocrit, which is a whole blood count. We'll then look at the volume. We'll look at the hemoglobin that's inside of the cells. We'll look at the hemoglobin that's inside of the average hemoglobin. We'll look at also the width of a blood cell, so we can see if it's macrocytic or microcytic. We do platelet counts. We'll then do a differential. This is typically automated nowadays, but sometimes in the olden days we used to do it manually. People would actually have to look at these things. And we'll talk about each of these different kinds of white blood cells in just a few minutes. And this will tell you the percent out of all the white blood cells, the percent that are each of the different kinds of white blood cells. And then the ABS is the absolute count in thousands per microliters. So we can actually count how many neutrophils there are per microliter. So how many thousands of neutrophils there are per microliter. So you can see this person was flagged for their platelet count being a little bit low. They were also flagged for their lymphocyte count being low and monocyte count being a little bit high. And so each of these values, when you begin to look at anything that's flagged, each of these values is key information at determining a disease that a person might have. So when you go to become a nurse or a physician's assistant, a doctor, you'll learn to read these values and then to understand the patterns of what diseases could possibly be because of the readings that you are looking at. Blood is a tremendously valuable piece of information that doctors, nurses, physician's assistants can use to determine what's causing a person's problem. So there's lots of information you can gather from looking at blood cells. Maybe one day you'll have a hematology class and you'll take a look at all these things under the microscope. But I just want to show you size variation here. Here's a microcyte. Here's a macrocytic cell. This is what a normal cell, the diameter of it, what it should look like. Sometimes you can have ovals. Sometimes you can have all different kinds of shapes. If you have too little hemoglobin, you can have different color variations there. There can be polychromasia, which is where you have different color variations inside. Each of these things represents a certain disease. So you could be shape variations. So you can be kind of, spherical with no donut hole. You can be oval. So you can be sickle shaped, or where you have like in this one, a burr cell is where you're shriveling up in the cell membrane. It almost looks like wavy edged. Okay, so we can look for inclusions that are inside of the cell. So, for example, parasites. We can see if blood cells are sticking together or if they're forming these rouleau complexes. So all kinds of things can be determined by looking at these cells under the microscope. Um, Okay, well, let's go ahead and talk about the ABO blood group. So we do have surface antigens. These are glycoproteins that are on the surface of our blood cells. At least some of us do. So you can have a surface antigen that we call surface antigen A. You can have a surface antigen that's called surface antigen B. You can have both those surface antigens and be called AB, or you can have no surface antigens, and that's what type O blood represents. Now, if you have surface antigen A inside of your body will naturally produce these disease-fighting chemicals called antibodies, anti-B. So what these antibodies do is they will target the antigen B protein. So if I were to inject into a person type B blood, if they have type A blood, these antibodies would stick to that blood and would agglutinate and cause the blood to stop circulating through the blood vessels. Okay, so it's very important that we blood type people before we give them blood transfusions. So if you have antigen B, that means you make antibody A. So you can't be given blood from a person with type A blood. People with antibodies with the AB blood produce both surface antigens, and therefore they don't make any antibodies, because if they did, they would kill their own cells. People with type O blood are going to have both antibodies, anti-A and anti-B antibodies. So you don't want to give a person with type O blood type AB blood, B blood, or A blood, but only type O blood. Okay, so people with type A blood, they're going to have the surface proteins that are antigen A. They're going to produce anti-B antibodies. They will be able to receive blood from type A blood and type O blood, and they will not be able to give blood to type B blood people or type AB blood people. Type B surface antigen is is on the surface of the blood cells of people with type B blood. They produced anti-A antibodies. They can give blood to type B and O blood people, but they cannot give blood to type A and AB people. So AB blood type has both surface antigens. They have neither anti-A or anti-B antibodies. They can give blood to everybody. They are the universal donor. But they really are not incompatible with anybody. They are compatible with all blood types. Excuse me. They are typically incompatible with all blood types. My apologies on that. You can't give type AB blood to A people, B people, or O people. So type O blood, it has neither surface antigen. They have both antibodies. So we can donate that blood, type O blood, to anybody since they don't have surface antigens, and but they are incompatible. They cannot receive blood from type A blood people, type B blood people, or type AB blood people. So the way we determine blood type is using anti-A or anti-B serum. This is the fluid portion of the plasma of blood collected from people that are that have these respective blood types or have these antibodies in their bloodstream. And so if we take untreated blood and treat it with anti-A serum, if the person's type A and they're treated with anti-A serum, that means they're going to have clotting that occurs. But if you take anti-B serum and give it to Let me see if I can get this straight now. So anti-A serum will actually destroy the blood cells, but anti-B serum will not destroy or bind to the A blood type. Therefore, the blood smear will look normal. So if I take a type B blood and use type B anti-sera, you can see that it agglutinates or sticks together. But type B blood given type A serum doesn't agglutinate. So we have just a normal blood smear there. So AB blood given either type of antibody will clump together, but type O blood does not clump given any of the antibodies because they don't have the surface antigens that the antibodies could stick to. Most people are always, kind of interested in what the blood types are for different groups of people. So you can just, as an example, you can take Black Americans. They have 49% of Black Americans are type O blood, 27% are type A blood, 20% are type B blood, 4% are type AB blood, and most of them, for the surface antigen called the Rh factor or surface antigen D, 95% of Black Americans are Rh positive. Okay, so you can just look through there if that's of interest to you. One small problem that we have, unfortunately, is there are 50 different kinds of blood surface antigens. One of them that's medically significant is the Rh blood group. So you may have heard people say I'm A positive or A negative. That negative or positive is the status of the Rh blood group. It's also known as the D blood group. And so one problem you have if you're a Rh negative mother, and it's relatively rare to be Rh negative, but if the mother's negative and she mates with an Rh positive male who sends the genes to build the Rh factor to the fetus, and the fetus produces Rh antigens, it's inevitable that a few of the blood cells from the fetus will cross over the placenta, that connecting organ between the fetus and the mother, and get into her bloodstream. Now, for the first pregnancy, it's not so bad, but unfortunately, because the Rh antigens go into the mom's bloodstream, she begins to produce antibodies to the Rh factor. So she's sensitized between the pregnancies. Second pregnancy, no bueno. What happens there are those antibodies, as the blood cells cross over from the second fetus, the antibody production is significantly ramped up. Those antibodies will travel through the placenta, and they will actually try to destroy the blood cells of the fetus that's inside the mother. This causes hemolytic disease of the newborn, or HDN, and literally it can destroy the blood cells and therefore cause a baby to die or be stillborn or have major problems at birth. Okay, so we have a solution for that. We can do blood testing for the mother. If the mother is tested as Rh positive, there's no problem. So if the mother's tested, she's Rh negative, then that could be an issue. RhoGAM and this is what RhoGAM comes in a package. It's injectable, and it's essentially anti-Rh antibodies that they extract from people, and these are injected into the female and who are Rh negative. And what these antibodies do is they will bind any Rh positive fetal cells that are in a mom's circulation, and what that does is since they bind to those Rh positive fetal cells, they don't allow the mother to react to the fetal cells, and therefore she doesn't have any immune reaction. She doesn't produce antibodies to the Rh factor. Now, these anti-Rh antibodies, there's not so many of them that they travel across the placenta and do damage to the baby. They're just enough to destroy whatever cells the fetus may be sending across the placenta so that the mom doesn't react to them. Okay, so in the olden days, sometimes an Rh negative female and Rh positive male would not ever have a viable child because of this problem with the Rh factor. Okay, I think I'll stop at this point, and we'll finish up with white blood cells, and we'll finish up with platelets and the stopping of bleeding in our next video. So until then, I will see you then.